animal organization. We were talking about the invertebrate phyla. We finished till Ctenophora. In Ctenophora, once again, the most important points I would like to put before you. Ctenophores are commonly called sea walnuts or gooseberries or raspberries. They are all marine, exclusively marine here now. They are also diploblastic. They are biradially symmetrical. In ctenophores, stinging cells are replaced by colloblasts or lasso cells. Colloblasts or lasso cells, which help in food capture. They secrete some adhesive substance, sticky substance that helps in food capture. Then they have about 8 comb plates or teens or costa in the body. 8 comb plates or teens or costa are present. They are meant for locomotion. That's why they are called comb jellies, They are all hermaphrodite. Fertilization is internal. Life cycle involves a larva called cydipid. The larva of ctenophores is called cydipid. Then most of them are bioluminescent. The only ctenophore with stinging cells is Achlora rubra. I made a mention already about this. The only stinging ctenophore with stinging cells is Achlora rubra. Then they have got 2 tentacles on the aboral side. Then it is divided into 2 orders. Ctenophora, phylum Ctenophora is divided into 2 orders. Order Tentaculata, order Nuda. As the name itself indicates, Tentaculata, 2 aboral tentacles are present with an aboral sense organ called statocyst. The examples for Tentaculata are Pleurobrachia, Harmiphora, Ctenoplana, Coeloplana. These 2 are commonly called comb jellies or sea walnuts or gooseberries And Ctenoplana and Coeloplana are the missing links between Cnidaria and Ctenophora. Apart from this, there is another example, Cestum. Cestum is commonly called venous girdle. Cestum is commonly called venous girdle. In Nuda, as the name indicates, tentacles are absent in Nuda. The example for Nuda is Barrow. Example for Nuda is Barrow. The next one we'll talk about now, phylum Platyhelminthes. The most important points which are likely to appear in the EAMCET, AFMC, JIPMER, AIMS, so on and so forth, I'll put before you. The term Platyhelminthes was coined by Guggenberg. The term was coined by Guggenberg. The term was coined by Guggenberg. And the Platyhelminthes it exhibits certain first formed characters. That is, characters which are not seen in the previous phyla, which make their appearance for the first time in Platyhelminthes are: number 1, triploblastic nature. So it happens to be the first formed triploblastic phylum. Number 2, it is a first formed organ system graded phylum. Organ system grade of organization is noticed. Number 3, for the first time, definite excretory organs are present. Definite excretory organs are present, because in Cnidaria and Ctenophora there are no definite excretory organs. In Platyhelminthes, for the first time, definite excretory organs are present in the form of flame cells. Flame cells happen to be the modified protonephridia. This is again important point. Remember, flame cells happen to be the modified protonephridia. The flame cells are concerned with excretion and osmoregulation. They are meant for excretion and osmoregulation. Then apart from this, ectolecithal eggs are present in them. Ectolecithal eggs, that is, yolk lies outside the egg, so ectolecithal eggs are present. Organ system grade of organization. Then another first formed character is, they are the first formed bilaterally symmetrical animals. They are the first formed bilaterally symmetrical animals. First time you find well-developed musculature in the body wall, like well-developed circular muscles and long-term muscles are noticed in the body wall. So that is also first form character, well-developed musculature in the body wall. Then the gonads are divided into germarium and vitellarium. Gonads are differentiated into germarium and vitellarium. All the gonads are collectively called germarium, and the reproductive glands which are associated with germarium are called vitellarium. So they are all first form character, triploblastic nature, organ system grade of organization, definite excretory system, that is in the form of flame cells which are meant for osmoregulation and excretion, ectolethal eggs, bilateral symmetry, well developed musculature, gonads distinguishable into germarium and vitellarium. Then apart from this, they are the first formed cephalized animals. For the first time, cephalization is also noticed in the platyhelminthes. Now, platyhelminthes, most of them are parasitic, a few are free living. Now, the platyhelminthes, ladder type of nervous system is present. Ladder type of nervous system is present. This is also one of the unique characteristic features of platyhelminthes. They have got ladder type of nervous system with ganglions. Ganglions also, they are ganglions in the form of cerebral ganglions. So the presence of cerebral ganglions is also one of the first form characteristics of platyhelminthes. Now the platyhelminthes is divided into three classes. Class Turbularia, class Trematoda, class Cestoda. Turbularia, Trematoda, Cestoda are the three classes of phylum Platyhelminthes. And Platyhelminthes happen to be the acoelomates. That is also the most important characteristic feature of Platyhelminthes. They are the acoelomates in which the body cavity is replaced by mesodermal tissue called parenchyma. Now the turbularians are free living. The outermost layer of the turbularians is syncytial ciliated epidermis. The outermost layer is syncytial ciliated epidermis. The epidermis is provided with certain structures called rhabdites. Rhabdites help in food capture by secreting mucus. Then in turbularians pharynx is irreversible. Irreversible pharynx is present. It starts protruding out through the mouth and they exhibit highest rate of regeneration. Rate of regeneration is high, and they are all free living organisms. Free living organisms. Turbularians are the free living organisms among platyhelminthes. Okay, then they have got a larva called, two larvae are there in this. The most important larvae of Turbularia are Muller and Gotes. Muller and Gotes are the larvae of Turbularians. And the most important examples for Turbularia are Convoluta, Planaria, which is also called Dugitia, Bipalium, Temnocephala. These are certain important examples of Turbularia here. In this, Planaria is also called Dugitia. Bipalium is the turbularian that lives in moist soil. Temnocephala is a connecting link between Turbularia and parasitic platyhelminthes. Trematodes are commonly called flukes. Trematodes are commonly called flukes. They are ectoparasitic or endoparasitic. Ecto or endoparasitic. They are ecto or endoparasitic. The outermost layer is tegument or cuticle. Tegument or cuticle. Intestine is bifurcated in them. Intestine is bifurcated in turbularians, in trematodes. Then they've got two suckers, oral sucker and ventral sucker. Oral sucker and ventral sucker. Again, most of the trematodes are with two suckers, and a few of them are with single sucker. It's again divided into monogenia with a single sucker, digenia with two suckers. Then they exhibit polyembryony. Life cycle involves many embryos which are considered as the larvae. Then all are bisexual. All trematodes are bisexual except schistosoma. Except schistosoma, which is unisexual. Except schistosoma, which is unisexual, all the other trematodes, which are commonly called flukes, are bisexual here. And all of them are parasitic. A few are ectoparasites and most of them are endoparasites. Now I'll give the most important examples now with certain unique characteristic features in the life cycle, which are the most important points for AFMC, JIPMER, AIMS, AAPMT and EAMCET. Polystomum. Polystomum is a fluke that lives in the urinary bladder of toads and frogs. It's an endoparasite. Then Diplozoan, Dactylogyrus, Gyrodactylus. They are ectoparasites on the gills of fish. Polystomum lives in the urinary bladder of toads and frogs. Diplozoan, Dactylogyrus, Gyrodactylus are the gill parasites of fish. Gill parasites of fish. Then the human parasites now. First you have the most important example, Schistosoma hematobium. Schistosoma hematobium is commonly called blood fluke of man. It's commonly called blood fluke of man. It is a digenetic parasite. Digenetic parasite with primary host is man. In human being, it lives in the blood capillaries of urinary bladder and pelvis region. Lives in the blood capillaries of urinary bladder and pelvis region. Primary host. The secondary host is snail that belongs to genus Bulinus. The secondary host is snail that belongs to genus Bulinus. Now the schistosoma, as I made a mention earlier, it is a unisexual parasite. Both male and female are separate. The females are stout and males are slender. In the females, on the ventral side, females are with a groove called gynecophoric canal. Females are provided with gynecophoric canal, in which the large males. So the females are stout and short. The females on the ventral side, they've got a gynecophoric canal that larges the male here now. Then the females, soon after fertilization, they lay terminal spined eggs. Females lay terminal spined eggs. Terminal spined eggs. These eggs come out along with the urine. Then on entering the water bodies, eggs hatch into larvae. These terminal spined eggs now hatch into larvae called miracidium. This miracidium swims in water in search of the primary host. Then when it comes in contact with a human being here now, that is people when they go for swimming in contaminated waters, or they start bathing or wading in contaminated waters, this Miracidium larva finds its way into the human blood vessels with the help of penetration glands, Miracidium swims in water in search of the secondary host, that is the snail. Now in the snail, the Miracidium transforms into sporocyst. The sporocyst in turn transforms into cercaria. Okay. So within the snail here now, the Miracidium larva transforms into sporocyst. The sporocyst in turn transforms into cercaria. The cercaria of schistosoma is provided with a forked tail. That's why it is called Furcocercus cercaria. Furcocercus cercaria. Now this Furcocercus cercaria, that is forked cercaria, comes out of snail and swims in water in search of the primary host, human being here now. In the life cycle of schistosoma, the two larvae are absent, that is redi and metacercaria. Redi and metacercaria are absent. Redi and metacercaria are absent. Now the cercaria or Furcocercus cercaria swims in water in search of the primary host, human being. When people swim in contaminated water or start bathing in contaminated water, this cercaria sticks to the skin somewhere in the ankle region or in the shank part or any other part of the body. Then with the help of penetration glands, it penetrates to the skin to enter peripheral blood vessels. From peripheral blood vessels, it reaches heart. From heart, it reaches lungs, Then from lungs, once again, it gets back to the intestine region, That is, it ascends the respiratory tract. From the pharynx, it enters intestinal blood vessels. From intestinal blood vessels, it reaches the blood vessels of pelvis and urinary bladder. Now that it completes the life cycle, And in the life cycle, you find only three larvae: Miracidium, sporocyst and cercaria. Redi and metacercaria are absent. Then these larvae, these eggs now hatch into Miracidium. The Miracidium, as I made a mention, enters into snail. Within the snail, it transforms into cercaria. The cercaria is infective to man. Cercaria larva is infected to man. Now, the cercaria, while penetrating to skin, it causes a kind of skin infection called swimmer's itch. So the larva, cercaria larva, while penetrating to skin, causes a kind of infection at the site of entry called swimmer's itch. Then in lungs and heart, they cause cardiopulmonary schistosomiasis. Cardiopulmonary schistosomiasis. Cardiopulmonary schistosomiasis in lungs and heart, Then these terminal spined eggs now come out along with the urine. While coming out, they rupture the blood capillaries. Sometimes they cause oozing out of blood through urine, which is called hematuria. Loss of blood through urine is called hematuria. Swimmer's itch, cardiopulmonary schistosomiasis and hematuria, all of them are put together called schistosomiasis. Schistosomiasis or bilharziasis. Schistosomiasis or bilharziasis. Okay, all the symptoms are put together called schistosomiasis or bilharziasis because schistosoma was discovered by Bilharz. So these are the important characteristics of schistosoma hematobium. Apart from this, there are another two species, schistosoma mansoni and schistosoma japonicum here. Now another example you have Clonarchis or Opistarchis. It is commonly called Chinese liver fluke or Oriental liver fluke. of man. It's commonly called Chinese liver fluke or Oriental liver fluke of man. It is a trigenetic fluke here now. Primary host is human being. First intermediate host is snail. Second intermediate host is fish. So primary host is man, first intermediate host is snail, second intermediate host is fish. So people get infected by consumption of contaminated fish, other fish which are deposited with larva, Cercaria larva. That is they are the important characteristics of Opistharchus or Clonarchus, which is commonly called Chinese liver fluke or Oriental liver fluke of man. We got another example, Paragonimus. Paragonimus is commonly called lung fluke of man. Paragonimus is commonly called lung fluke of man. It is also a trigenetic trematode. Primary host is man. First intermediate host is Cyclops or Diaptomus. Second intermediate host is crab or crayfish. First intermediate host is Cyclops or Diaptomus and second intermediate host is crab or crayfish. So people get infected by consumption of contaminated crab or crayfish. And it is also a trigenetic parasite here with three hosts commonly called lung fluke of man. Another example, Fasciolopsis busci. It is the largest intestinal fluke of man. Largest intestinal fluke of man. Largest intestinal fluke of man is Fasciolopsis busci. Another most important example for trematoda is Fasciola hepatica. Fasciola hepatica. Fasciola hepatica is commonly called liver fluke of sheep. Liver fluke of sheep. It lives in the bile ducts of liver of sheep. It is a digenetic parasite. Primary host is sheep. Secondary host is snail that belongs to genus Limnia. Snail that belongs to genus Limnia. The unique characteristic feature of this parasite is the parasite has a vestigial vagina called Laurer's canal. Vestigial vagina called Laurer's canal is present. Is important. It can be asked in AFMs, JIPMAR, or AIMS, or any other exam. Laurer's canal is a vestigial vagina that makes its appearance at the time of fertilization in Fasciola. And unlike other trematodes, this particular trematode has got all the 5 characteristic larvae. That is, egg hatches into larva called Miracidium. The Miracidium enters into snail. Now inside the snail, Miracidium transforms into sporocyst. The sporocyst transforms into red eye. The red eye transforms into Cercaria. The Cercaria transforms into Metacercaria. Metacercaria enters the primary host, sheep. Okay, so this is the life cycle here now. The eggs come out from the sheep here now. They hatch into Miracidium larva. The Miracidium enters snail. Within the snail, miracidium transforms into sporocyst. Sporocyst gives rise to red eye. Red eye gives rise to cercaria. Cercaria gives rise to metacercaria. Metacercaria comes out of snail and swims in water in search of the primary host. Then when the sheep go for drinking, suppose they happen to drink contaminated water, then metacercaria enters sheep, where it transforms into adult. Then this fasciola hepatica causes liver rot in sheep. It causes a disease called liver rot in sheep. Now we'll talk about the class Cestoda, which are commonly called tapeworms. All cestodes are commonly called tapeworms. All cestodes are endoparasites. In all tapeworms, gut is absent. Gut is absent because they feed on pre-digested food. They exhibit pseudometamerism. Body is divisible into scolex, neck and strobila. Scolex, neck and strobila. The outermost layer of the body wall is syncytial tegument. Outermost layer of the body wall is syncytial tegument. And all tapeworms are bisexual except Diecocestus, which is unisexual. Except a tapeworm Diecocestus, which is unisexual, rest of the tapeworms are bisexual. The important examples for the Cestoda I shall do. The most important characteristic features of each parasite I shall put before you. We'll start with first Taenia solium. Taenia solium belongs to class Cestoda, subclass Eucestoda. Subclass Eucestoda. Once again, class Cestoda is divided into two subclasses, Cestodaria and Eucestoda. Cestodaria and Eucestoda. In Cestodarians, body is not divisible into scolex, strobila and neck, whereas in Eucestodes, body is divisible into scolex, neck and strobila. So it belongs to Eucestoda. Okay, then order Cyclophilidae. Order Cyclophilidae. Now it is commonly called pork tapeworm of man. Pork tapeworm of man. It is a digenetic parasite. Digenetic parasite. Primary host is man and secondary host is pig. And people get infected by the consumption of undercooked measly pork. They get infected by the consumption of pork infected with the larvae called measly pork. Now this scolex is radially symmetrical with a rostellum. It has got a rostellum, then two rows of hooks, about 22 to 32 chitinous hooks are present. 2 rows of 22 to 32 chitinous hooks are present. It has got a protrusible structure called rostellum and 4 suckers, which are also called acetobula. Suckers are also called acetobula. The main function of the scolex is adhesion. That's why it is also called hold fast organ. Scolex is also called hold fast organ. So this is the rostrum, which is a protrusible structure here now. At the base of rostrum, there are hooks here now, and these are suckers. 4 suckers are present. So this is rostrum, which is meant for adhesion. That's why it is called hold fast organ here now. Then the rostrum, that is scolex, is followed by neck. Neck acts as a growth zone. It gives rise to proglatids by transverse fission called strobilization. It produces proglatids at the rate of 2 to 8 per day by a process called strobilization. Then the strobila, it contains about 800 to 1000 proglatids. 800 to 1000 proglatids here now. Then strobila in turn consists of immature proglatids. Immature proglatids which are broader than long. Mature proglatids which are squarish. Then gravid proglatids, gravid or ripe proglatids which are longer than broad. Immature proglatids, broader than long, about antimost 200 proglatids are immature here now. Then the mature proglatids, the antimost say 300 to about 650th are mature proglatids. Among these, the first 100 mature proglatids are with mature testis and immature ova because of protandry nature. Then the rest of the mature proglatids, that is say 300 to 650th, are the mature proglatids with both testis and ovary. Then the mature proglatids, upon fertilization, they transform into gravid or ripe proglatids. They are longer than broad. Then the mature, once gravid proglatids are formed in large number, they get detached from the parent and come out along with the feces for dispersal. The phenomena of the detachment of proglatids now to ensure dispersal and propagation of the parasite is called apolysis. So the parasite undergoes apolysis to facilitate dispersal of the parasite, and it also helps in restricting the length of the parasite here now. The body wall consists of outer tegument, the middle musculature, and inner mesenchyme. So the body wall consists of tegument, musculature and parenchyma or mesenchyme. So this is the body wall of Taenia solium here now. This entire zone can be identified as tegument. The outermost layer, this is the outermost layer of tegument here now. The outermost layer of tegument is called comedial layer, which is provided with numerous microvilli, hair-like projections called microvilli. Below comedial layer, it has a homogeneous layer. Below that, it has got a basement membrane. Then below tegument, that is, tegument consists of comedial layer, homogeneous layer, basement membrane. Below tegument, it has got musculature. This can be identified as musculature. The musculature consists of outer circular muscles and inner longitudinal muscles. Longitudinal muscles. Then the rest of the thing can be called mesenchyme. The mesenchyme is provided with parenchyma, syncytial parenchyma, and the glands. There are certain glands which open into tegument. They are called subcuticular glands. Subcuticular glands open into tegument. They give rise to tegument. And certain glands open outside. These glands now, they secrete anti-enzymes. They open outside through minute perforations. Those glands which open outside through minute perforations, they secrete anti-enzymes to destroy the host enzyme so that the Taenia solium remains unaffected by the enzymes of the human gut. The parenchyma and along with the glands can be called mesenchyme. The body wall consists of number 1 tegument, number 2 musculature, Number 3, mesenchyme. Then the mature proglotid, if you look into the structure of mature proglotid, it has got about 150 to 200 testes. 150 to 200 testes are present with vasa efferentia. All vasa efferentia unite to form vas deferens. That vas deferens opens outside through genital atrium, through male genital pore. And if you take the female reproductive system, it has got a bilobed ovary. Bilobed ovary. Those two lobes are connected to each other by ovarian bridge or ovarian isthmus. And there is an uterus with uterine duct. And there are two reproductive glands here now. One is vitelline gland. Vitelline gland. The vitelline gland lies below the ovary here now. Vitelline gland secretes yolk cells and eggshell. Vitelline gland secretes yolk cell and eggshell. Okay. Whereas there is another gland called Michaelis gland, or it is also called shell gland. It is there around the ootype. Okay, this Michaelis gland or shell gland secretion lubricates the passage of egg capsules from the ootype into the uterus. From the ootype into the uterus. So the mature proglotid consists of 150 to 200 testes with vasa efferentia and vas deferens. And in the female reproductive system, you find a bilobed ovary. Both the lobes are connected to each other by a transverse ovarian bridge or ovarian isthmus that in turn gives rise to a median oviduct. Oviduct opens into ootype. Around the ootype, there is a Michaelis gland or shell gland whose secretion lubricates the passage of egg capsules into the uterus. And below ootype, there is a vitelline gland secretes yolk cells and eggshell. It secretes yolk cells and eggshell. The zygote fertilization takes place in the ootype, and the zygote surrounded by yolk cells and eggshell is called egg capsules. So all these egg capsules from the ootype find their way into uterus through uterine duct, undergo further development in the uterus before the apolysis could occur. The zygote, by unequal holoblastic spiral determinate cleavage, it gives rise to micromeres, mesomeres and megameres. The micromeres give rise to the morula, that is embryo proper. The mesomeres give rise to protective layers, two hexacanth membranes and an embryo 4. The megameres, along with the yolk cells they form outer embryonic layer. Zygote undergoes cleavage to form micromeres, which give rise to the embryo proper morula. The mesomeres give rise to two hexagon membranes and an embryo 4 or secondary eggshell. The megameres and the yolk cells put together form outer embryonic layer that nourishes the developing zygote. So if you look into this, it is the morula that transforms to embryo. It is provided with 3 pairs of hooks. And a pair of penetration glands. Then it is surrounded by 2 hexagon membranes. And there is so this complex structure is called oncosphere. This oncosphere contains centrally situated hexacanth, which originates from the morula. Hexacanth is provided with 3 pairs of hooks and a pair of penetration glands. Hexacanth is surrounded by 2 hexacanth membranes and an embryophore. The embryophore is also called secondary eggshell. Embryophore and hexagon membranes originate from mesomere, and it is surrounded by an outer embryonic layer. This outer embryonic layer is formed by fusion of megameous and yolk shell, and the outermost is the eggshell. Outermost is the eggshell. That eggshell is a contribution of vitelline gland. So this entire complex structure is called oncosphere. Soon after the formation of oncospheres, apolysis takes place, so that the oncospheres come out of human being along with the fecal matter, and they enter pig hen. Then inside the pig hen, these oncospheres lose the outer layers, they become naked hexagon. The naked hexagon penetrates to the gut wall of the pig and reaches voluntary muscles of thigh, shoulder, ham, tongue, neck. Then on entering the voluntary muscles of the pig hen, it undergoes certain changes to form a larva called cysticercus cellulosae. The cysticercus cellulosae remains hidden within the muscles of the pig hen. It remains alive for about 5 to 6 years, and the muscles deposited with cysticerci are collectively called measly pork, and when the people eat undercooked measly pork, they get infected. That cysticercus cellulosae, on entering the intestine of man, it undergoes evagination to form the young one. The young one ultimately transforms into adult. So the infective stage of Taenia solium to man is a measly pork deposited with cysticerci, and infective stage to pig is the oncosphere. Then the adults cause teniasis, which is characterized by various gastrointestinal disorders. Then the larvae caused cysticercosis, which is more fatal than teniasis. The larvae, sometimes they may enter the vital organs of the human body like eyes, brain, heart, kidney, lung and all that, where they undergo, they get calcified and thereby they form cysts. So that proves to be more fatal than the symptoms caused by adult.